Hello students, this is Faizan Mirza. We are discussing dihybrid cross today. Dihybrid cross means we discuss inheritance of two traits at the same time, and for now, we'll just try to imagine that the two traits that we are discussing are following independent assortment. They are not linked, and there is no linkage. The two traits that we are discussing today are plant stem color and the leaf shape. Plant stem color, the first trait could be either the purple stem or green stem. Purple stem is having a dominant allele capital A and the green stem is small a recessive allele. So there are three combinations of the genotype capital A, capital A, capital A, small a. Both will be purple since they have the capital A allele. And small a will be green because it is having a homozygous recessive condition. The only condition which the green color will be present in the phenotype. The leaf shape is the second trait. Cut leaf is capital D and potato leaf is small d. You can have again three combination capital D, capital D, capital D, small d. Both will be cut leaf, and the only possibility for leaf to be potato or rounded will be small d combination. What are we doing here? We are trying to just see that whether the two into traits are following independent assortment or not. So the possible combinations could be these, which are listed here. The you can see four different type of phenotypes that can be found. The plant could either be purple stem with cut leaf, the plant could either be purple stem with potato leaf, the plant could be green stem with cut leaf, and could be green stem with potato leaf. So four different combinations can form in various possibilities, and these are the various genotypes that are listed. As far as there is a single capital A dominant allele, the stem color will be purple. As far as there will be a capital D, the leaf shape will be cut. For the green stem to show itself, the recessive state of AA must be present. For the potato leaf, small d must be present together. So you can just relate these relevant genotypes with their phenotypes. So the first cross to perform when we are discussing that the genes are not linked is that you cross the purebred varieties of both the traits together. So you cross purebred varieties of both traits together. Purebred basically means that they are homozygous conditions. So homozygous condition means that you take homozygous for both traits, purple stem and cut leaf, capital A, capital A, capital D, capital D, and you cross them with green stem and with potato leaf, that is small a, small a, small d, small d. Both are homozygous dominant and homozygous recessive respectively for both the traits. The gametes will follow in meiosis, and you will have two kind of gametes being forming, one for each, all capital A and capital D gametes from for the first parent, and all small a small d gametes from the second parent. Now when you cross them together, you see that all the offsprings of the F1 generation will be purple stem and cut leaf, and they all will be hybrid, capital A small a, capital D small d. 100% offspring will be hybrid, and since the dominant allele for stem color as well as leaf shape are present, so the dominant features will be expressed, but the offspring will all be hybrid. What do we know do next is we cross the offspring from the first generation and cross them with the purebred recessive for both the traits. Here the purple stem cut leaf offspring from the F1 generation, capital A small a, capital D small d, is crossed with the green stem potato leaf, the homozygous recessive for both the traits, the parent of the first F1 generation. The gametes formed by the homozygous recessive will be all small d as done previously, but the stem, purple stem and cut leaf, capital A, capital D, small d, can form four different kind of gametes. And since the gametes are following independent assortment, a dominant allele capital A can come with a capital D. Capital A can come with a recessive D. A recessive A can come with a dominant D. A recessive A can come with a recessive D. So there are four different kind of combinations which can form in the gametes. It's not possible for two capital A's or two capital D's or two small d's or two capital D's to come together in a gamete, because in meiosis the homologous chromosomes separate. Only one of each allele ends up in a single gamete. So when you cross them together, you perform this cross. You see that there are four different outcomes, and there is an equal probability of all four forming. You will have purple stem cut leaf. You will have green stem potato leaf. This one is similar to this parent, and the green stem potato leaf is similar to this parent. Whereas there are two varieties, purple stem potato, which basically is a new variety. Then there is a green stem cut leaf. This is also a new variety. Both these phenotypes were not present in the either parents. These are the new varieties which are also called as recombinants. So the chances of all the four forming was 1 is to 1 is to 1 is to 1. This gives you an idea that yes, probably the genes here, which is the gene for the stem color and leaf shape, were following independent assortment. What do you mean by independent assortment? That the homologous pair was capital D small d, another homologous pair was capital A small a. There was an equal probability of the dominant allele A coming with dominant allele D, recessive allele A come in with dominant allele D, or a dominant allele A come in with recessive allele D, and a recessive allele A and a recessive allele D coming together, there was an equal chance there. The genes are not linked. To be certain about the independent assortment, a third cross can be performed. The crossing of the offspring, the first F1 generation together. You have purple stem and cut leaf offspring. You cross it with another of its own kind. Both will be capital A small a, capital D small d. And again, the same condition in the other. That these are genotypes. The gametes will be four of different kinds. The dominant A and D is a dominant and a recessive AD, a recessive and a dominant AD, both recessive AD. You place them on the square. You draw four boxes, and this time the four boxes will be actually 16 squares, so you have 16 squares each. You just cross each and you place them together. Since the genes are not linked, we'll be placing them just in the order of alphabetical order that we'll follow, and we'll place the dominant allele first. So AADD when crossed together will form capital A, capital A, capital D, capital D. The next will have capital A, capital A, capital D, small d. The third one will be capital A with a small a. So the capital A here comes from this parent, and the small a comes from this one. The capital D comes from this, and the capital D comes from this. So this again can be formed. So this is how you just fill this up. For example, if you just let's talk about this. This is how this coming about. The small a from here coming here, and the small a from this gamete landing here. The capital D, the dominant D is coming from here, and the dominant D coming from here. So this is how you just fill up all these. This is, for example, this is small a, small a, small d coming together. You just complete the cross with the letters first, and then you start to fill up the phenotypes of each. Wherever at least a single capital A was mentioned, you know that this is the dominant allele for the stem color. So you just mention the purple color of the stem. And wherever you have at least a single capital D, at least a single capital D means the leaf shape would be cut. So you just mention purple stem, cut leaf for each. You will, when you place them together and you count them, you will see that purple stem and cut leaf are 9 in count out of these 16. And then when you count purple stem and potato leaves together, they will be coming out to be 3. Then when you count the green stem and cut leaf, again, they will be coming out to 3. And the green stem, potato leaf, this last one will be only 1. So the ratio of 4 different possibilities coming together will be 9 is to 3 is to 3 is to 1. If you're getting this ratio, this means that the two genes which you are discussing, capital A small a and capital D small d, they are sort independently in meiosis 1, and the genes of stem color and shape of leaf are not linked together. If they were linked together, the new recombinant would not have formed, new variety would not have formed. 9 is to 3 is to 3 is to 1 ratio in this cross would not have occurred, and the ratio of 1 is to 1 which we discussed here would not have occurred either. So this actually tells you that yes, genes for stem color and the leaf shape are following independent assortment, and they are not linked together. That's it. Thank you.